Hello and welcome back to your Chem 4 lecture. Today we're going to move on in chapter 17 and talk more about the structure and function of both DNA and RNA as nucleic acids, that is genetic storage, and then how it's used for encoding proteins, basically. But let's start by talking about the structure. So this is the famous double helix model. We're going to talk about how that was determined in this lecture today. So in the 1940s, there were certain things that were known about DNA. So biologists noticed that the amount of adenine bases was equal to the amount of thymine bases. And likewise, the amount of guanine bases was equal to the amount of cytosine bases. So it seemed like at the time, there was some kind of base pairing going on, where adenine always paired with thymine and guanine always paired with cytosine. So basically, you just had the overall structure here that the number of purine bases equaled the number of pyrimidine bases, because the number of adenine equaled the number of thymine bases, and the number of guanine equaled the number of cytosine bases. So A equals T, G equals C. So some base pairing was going on. But it wasn't until there was more structural information about DNA that we were able to understand why this was and how this could be. So the people who developed the model for the double helix of a DNA are James Watson and Francis Crick. They built on the work of Rosalind Franklin, a very talented crystallographer, who took the first photographs of a crystal structure of DNA that allowed for the visualization that led to the realization that there was a double helix structure. So the double helix has these two polynucleotide strands, and they wind around each other sort of like a spiral staircase. The sugar and phosphate forms a backbones, and that's like the outer stair railings, or kind of like a ladder if it's curving. And then the bases are the steps along the inside. Now, interestingly, one strand is going to go from the 5' to 3' direction, and then the second strand basically goes the opposite way, the 3' to 5' direction. So if you're kind of counting it, it's like they go opposite ways in a way. But they always pair in this way. An adenine base always pairs with a thymine base in the other, and a guanine base always pairs with a cytosine base in the other. Now, the reason this works is actually chemistry, okay? So the reason that they pair in this way, and they pair specifically this way, is that adenine has two hydrogen bonding locations. It has a hydrogen off of the amine group up here that can hydrogen bond to the carbonyl on thymine, and it has a nitrogen that can accept a hydrogen bond from the protonated nitrogen on the thymine. So you see they line up exactly this way. This one gives a hydrogen bond, this one accepts a hydrogen bond. It wouldn't work if they were paired with anything else. Only guanine and cytosine kind of pair the opposite way, where guanine has a carbonyl oxygen that accepts a hydrogen bond, then a nitrogen that gives a hydrogen bond to a nitrogen on cytosine, and then a nitrogen that again gives a hydrogen bond to another carbonyl on cytosine this time. So this matching basically allows for base pairing, where because of these hydrogen bondings, you can't fit a guanine next to a thymine or an adenine. It can only pair with a cytosine, and it can't pair with itself because, again, it's taking, giving, giving, whereas here, cytosine is giving, taking, taking from hydrogen bonds. So it's like a key, basically. And only those two bases will fit with each other. Okay, so again, the double helix has these two strands of nucleotides. They wind together, and they're held together by the two hydrogen bonds that form between adenine and thymine, and then 3 hydrogen bonds from guanine and cytosine. And this kind of gives you an idea of what that looks like here. So again, remember the 5' end of one strand is going to be matched by the 3' end of the other. And that's what gives the geometry for this DNA double helix. Okay, so given that you know about base pairing now, let's look at our sample exercise packet and write a complementary base sequence for the following strand in the DNA section. And let's say that we're starting from the 5' end over here and ending at the 3' end over here. So I give you the sequence AGT, CCA, ATC. What is going to be the complementary sequence on the corresponding strand of DNA. Pause the video when we take a look at the answer when we come back. So pause. Okay, so A pairs with T, G pairs with C, T pairs with A, C, G, C, G, A, T, A, T, T, A, C, G. So it ends up being TCA, GGT, TAG. If you have any questions on that, please make sure to answer in the comments below. Again, we're just kind of pairing up opposite strands of DNA. Okay, so now why is DNA so important to our cells? Well, DNA is really important because the information that it encodes allows for the proteins to be synthesized that make up all of our biochemistry. And the reason that DNA is used in this way is that it has two separate strands. And these strands can be unwound. And after they're unwound, then they can be replicated against each other using complementary base pairing. This is exactly what happens in DNA replication. The strands in the original, what we call the parent DNA molecule, are going to be separated. So you can break those hydrogen bonds apart. And then that allows a protein, a replicating set of enzymes, to come in and make new complementary DNA strands to the individual strands. So whereas you had one strand, you unzip it, make the complementary strand to both strands. Now you have 4 strands. Okay, so let's talk about this process of replication. First, an enzyme called helicase. It does exactly what it sounds. It unwinds the double helix, basically breaking the hydrogen bonds between complementary bases. Then we have, single strands that are open and exposed to a DNA replication enzyme. So basically, helicase unwinds it, and then we have DNA replication occurring here on both of the daughter strands here. So this is the parent DNA. It gets unwound, and we form 2 new strands based off of the 2 original strands from the parent DNA. So what ends up happening is you get 2 perfect copies of the DNA. So complementary base pairs are put into the location, and DNA polymerase basically allows or catalyzes the formation of the phosphodiester bond. So this way, the entire double helix of the parent DNA is copied into 2 new DNA strands. They're identical to each other, and they're exact copies of the parent DNA. So this is obviously super important. It's think about like when you're writing down your notes. If you wrote something down in your notes wrong, then when you took a look at those notes on the test, you would have the wrong information on the test. Here, we're talking about whether or not we get cancer. So like if we had some issue with our replication, then all of a sudden, one cell would be totally different from another cell, and it'd be very difficult for life to kind of self-reproduce. Because of DNA and the base pairing aspect of DNA in particular, information can be passed along from cell to cell and then from parent to daughter in exactly the same way. So this is kind of the function of nucleic acids here. DNA in particular is for the storage of this information. Okay, so looking back at our sample exercise packet, how many hydrogen bonds link each of the following base pairs? You have adenine, thymine, and guanine, cytosine. So it's a different number for each is what I'll tell you. Pause the video, take a look at your notes, and when we come back, we'll take a look at the answer. Okay, so for A and T, those are linked together by 2 hydrogen bonds. G and C are linked by 3 hydrogen bonds. Okay, so now that covers DNA. Really, DNA, what we want to do is we want to understand DNA can be copied using the replication enzymes that we talked about. But how does that relate to RNA? And RNA is very closely related to DNA, as we've talked about. So let's go ahead and take a look. So while DNA is kind of the most commonly referred to nucleic acid, maybe in popular culture, RNA is actually pound for pound the most abundant nucleic acid in the cell. And the reason for that is because RNA takes genetic information from DNA and takes it to the place where it needs to be read in order to produce new enzymes and new proteins. So yeah, this is basically the transmitter of the genetic information so that day-to-day cell operations can occur. So here's the difference between RNA and DNA. Remember that the sugar in RNA is ribose compared to deoxyribose for DNA. Uracil takes the place of thymine. And then the big difference is that RNA molecules are single-stranded. So they only basically are one line of RNA. They don't base pair or form a double helix in the same way as DNA. And also, RNA molecules are much smaller than DNA molecules. So your DNA strands tend to be very long. They make up your chromosomes. But RNA molecules are just very small segments of that DNA that are only used, to kind of transmit that genetic information to where it needs to go in the cell. So there's three main types of RNA that we're going to talk about. We have messenger RNA or mRNA. We have transfer RNA or tRNA. And then we have ribosomal RNA, which is known as rRNA. And so there's these three different types, and they each have their own function. So mRNA takes the genetic information from DNA and takes it to the ribosomes that are in the cytoplasm. This is where translation occurs. Transfer RNA is a particular shape of RNA. We're going to take a look at it here more closely in a minute. But what it does is basically works with mRNA to take the mRNA message from the DNA and turn it into specific amino acids in a process known as translation. That's transfer RNA. And then ribosomal RNA is basically is used in combination with proteins to form ribosomes. So if you've had your cellular biology class, you may know that ribosomes are the place or the location where proteins are made, manufactured, translated. So RNA basically functions in all of these ways to work with ribosomes to produce proteins. Okay, so as a total percentage of RNA, ribosomal RNA makes up about 80% of it because you're constantly pumping out proteins, just nonstop. Messenger RNA makes up about 5%, and that's because messenger RNA can be kind of read again and again and again until it decomposes. And then transfer RNA is used in these same reactions as far as translation goes to bring specific amino acids to protein synthesis. And that makes up about 15% of the total RNA in your cell. Okay, so I mentioned tRNA has a particular structure. Now, I'm not going to ask you to draw this here necessarily, but just a few things to note. It's a clover leaf structure, and then there's two important locations here. At the stem of the clover leaf, we have this what's called an acceptor stem. And this acceptor stem is basically going to be where the relevant amino acid is going to be placed during translation, because this is all to do with protein synthesis, so we need amino acids. The other part is at the very bottom of the middle clover here, I guess you could say, we have what's called an anticodon. And what this is three bases. So again, that's, A, G, U, and C. Anticodon. And what these do is they act as complementary bases to three bases on messenger RNA. So basically, three bases on mRNA are going to code for a particular anticodon, and that particular anticodon will carry with it a particular amino acid. And that's what's going to give us the primary structure of our peptides and our proteins. So here we go. Protein synthesis. This is the big one. This is what we want to talk about the most. We have two separate processes. We have transcription and we have translation. Transcription occurs in the nucleus, and this is where DNA is transcribed into messenger RNA. That mRNA is then taken to the ribosomes, and that is where translation occurs. This is where mRNA is translated by transfer RNA into amino acids. The particular amino acid sequence works to produce a protein. We'll talk more about this in a little bit. But yeah, so transcription, again, that's in the nucleus. Translation, you'll talk more about this, like where it occurs in your biology class. For now, we'll call it the cytosol, but a lot of times it happens at like endoplasmic reticulum, the rough endoplasmic reticulum in particular, but that's not part of this class, so don't worry about that. Okay, so here we go. Now we're ready for transcription. So this is when DNA that contains a gene unwinds and an RNA polymerase enzyme So yeah, transcription is where we're taking DNA, turning it into RNA. And we have RNA polymerase. RNA polymerase is exactly what it sounds like. It basically synthesizes RNA from a template strand of DNA. So mRNA just uses the same complementary base pairing that we did with the DNA, only uracil pairs with adenine. So instead of thymine, we have uracil, which we already talked about here. So for example, here, if we have the DNA template strand that's ATG, CTA, GGC, we know that it's going to pair A to U, T to A, G to C, C to G, T to A to U, G to C, G to C, and then C to G. This is kind of as the mRNA is being synthesized, what we're seeing here. So RNA polymerase, again, we're not going to take too much of a look at these enzymes here. We can just think of them as a couple of big blocks here. And all that's doing is basically moving along the DNA in the same way that DNA polymerase did in replication. And all it's doing is making this messenger RNA that can then be taken to the ribosomes for translation. Yeah, so then at some termination site, RNA polymerase ends, and that's where you stop making the mRNA, and that's where your mRNA is going to get cut, and that's where it is then taken to translate. Okay, so here we go. Let's practice our base pairing again. What is the sequence of bases in mRNA produced from the section of the template strand of DNA that has a sequence of bases CTHG, the mRNA sequence that matches up to it? Go ahead, pause your video now, take a look at the answers, try to come up with what it's going to look like, and we'll take a look at the answers together when we come back. So pause. Okay, so what's your base pairs, right? You know that cytosine pairs with guanine, so it's got to be A or B. It can't pair with itself, so that's not it. Thymine pairs with adenine, so adenine and adenine. Now, adenine pairs with thymine and DNA, but we're talking about mRNA here. So we know that rather than thymine, we have uracil. So it's got to be A pairs with U, A pairs with U, G pairs with C, G pairs with C. That's the only thing that makes sense for transcription. So the correct answer here has to be B. And that makes sense for our base pairs. Okay, that does it for transcription. In the next lecture, we're going to talk about the genetic code, what it means, and how it can be modified. So I'll talk to you soon. Until then, let me know in the comments if you have any questions. Bye-bye.